Hello, my name is Michael Cramer. I'm the Assistant Director at the University of Notre Dame Environmental Research Center, or UNDERC, and I'll be talking to you today about the effects of predation cues on the foraging behavior of woodland deer mice. Predation is a ubiquitous but complicated multi-species interaction that can have a variety of demographic as well as behavioral effects, and these behavioral effects are becoming better understood using the concept of the ecology of fear. Basically, prey perception of predation risk can fall into two different types of cues. Direct cues, which indicate the presence of an actual predator and would require an immediate response, and indirect cues, which are more generalized risk or habitat-based and are more longer term. The question we're asking, how do foragers respond to these different cues of predation risk? And we investigated the behavioral response using concepts derived from optimal foraging theory, specifically search and handling time. Woodland deer mice, or Peromyscus maniculatus gracilis, provide an excellent model system to address these types of questions. They are prey to a variety of predators that utilize different hunting strategies. They occupy a central position within the trophic food web, being primary consumers of seeds and prey for secondary consumers. And as seed predators, they also have wide-reaching effects on tree establishment and forest composition, especially in areas like where we work, which is on the border of Wisconsin and the Upper Peninsula of Michigan. They're also easy to capture and delightful to work with in the laboratory, so we can take mice from the forest into the field and address some of these questions about foraging behavior. Our main question is, how do predator cues of direct and indirect affect the foraging behavior in deer mice? So the direct cue indicates the actual presence of a predator. And so in these instances, mice should curtail their foraging to reduce a predator attack. And again, these behaviors can be specific responses to particular predators. But in general, they should move less. So they should increase their or decrease their search time and take more time to open seeds or increase their handling time. And instances where they have an indirect cue, such as substrate, which might affect general safety, they should forage less in these loud substrates that might attract the attention of a predator that is just happening by, and provide a generalized response for any predator. And so the predictions are that in a loud substrate, you would see the similar things to what you see in the direct cue, where the mice would maybe not move less, but move more carefully, and again, increase their handling time so that they make less noise. Our experimental design was set up looking at three factors. We had two seed types, sugar maple and red maple, which were determined by weight. So we put 10 grams of each seed type for the various trials. Our owl treatment was barred owl call versus trials where there was no owl call at all. And then we tested on different substrates, in this case sand versus leaves. And the leaves were artificial leaves that were made out of paper. Mice were captured in the forest and then kept in the lab. Trials were run for 30 minutes after an acclimation period that was determined by the mouse itself. So once the mouse started to forage, that's when the trial began. The arena was lit with infrared lights so that we could videotape them. And then those videos were scored for search and handling time with the program JWatcher. And for statistical analyses, we did an analysis of variance using search time per bout and handling time per seed as dependent variables, and all data were log transformed to make them normally distributed. For search time, there was a significant effect of owl treatment in that search time was higher when there was no owl call. So that is consistent with kind of what we were thinking. However, for the indirect predation cue, it was less clear. There was an interaction between the maple seeds and the substrate. So for sugar maple, the search time was higher in the leaves than it was for red maple, or than it was in the sand. And for red maple, there was no effect of substrate at all on search time. And if you look within the leaf trials, the search time for sugar maple was also higher in the leaves. Handling time was a lot more complicated. There was a significant three-way interaction between maple seeds, owl treatment, and substrate. So if you break it down, handling time increased in the red maple in leaves with the owl present. So in the presence of both predator cues, The response to by the mice for red maple was to increase their handling time. There was actually no effect on handling time for sugar maple. The handling time was about the same regardless of the different predator cues. But in the leaves, the handling time was higher for sugar maple than red maple in the leaves in the absence of owls. So key findings for search time was that mice always searched for seeds longer in the absence of a predator, and that could reflect time budget. They were more vigilant in these dangerous habitats, although it only was for direct cues of a predator. In the loud substrate, the handling time the search time was the same. And we also found no evidence of differences in other anti-predator behaviors, such as overall movement or freeze behavior. There could have been complications for the search time due to seed size. As I said before, sugar maple seeds are much larger than red maple seeds, and since we did it by weight, if we put in 10 grams of red maple seeds, there'd be a lot more seeds than when we put in 10 grams of sugar maple seeds. And there's also the possibility that red maple seeds may have been obstructed by the leaf litter, but that wasn't supported by the analysis because there was no effect of leaf litter on the search time for red maples. The characterization of the search time could have been inaccurate or problematic. The assumption that the number of bouts equals the number of seeds consumed was not held up, and that there were differences in the search time between the seed types was not consistent with the consumption data, which showed that the mice ate the same amount of sugar and red maple seeds. So the mice may have been finding the seeds and not consuming them, or they may not have been searching for seeds at all. They may have been searching for something else, like an escape route. So we need to find a better way to estimate the search time. For the handling time, sugar maple handling time was always higher than red maple, except in trials where there were owl calls. And so for sugar maple, there was no difference in handling time at all with either of the indirect or direct cues of predation. And the mice increased their handling time for red maple, which was interesting and might reflect two different strategies. In a risky habitat where there's no immediate predator, the strategy might be to open the seeds quickly in case a predator shows up. And in a risky habitat where there is an immediate predator, they open the seeds slowly so you don't attract any more attention to your activities. And why did we not see the same thing for sugar maple? Not sure. Could have been there wasn't enough. It took too much effort to gnaw through the husk, and then mice might have abandoned these seeds in high-risk areas and been a little more preferential. So both Marie Tosa and Daniel Osborne were students in the UNDRIC program and did most of the work. We obtained funding through the University of Notre Dame College of Science and also through the Bernard J. Hank Family Endowment. And if you have any questions, you can DM me on Twitter at @gsobrinus. Thank you.